Welcome to the Explainer. Today, we're grabbing our markers and heading to the whiteboard to literally break down the deadliest viruses on the planet, right down to their molecular architecture. We're going to demystify exactly how these microscopic assassins actually operate, piece by piece.
So here's our game plan for today. We'll hit Section 1, Anatomy of a Virus, Section 2, Mechanisms of Infection, Section 3, Metrics of Deadliness, and finally Section 4, the deadliest viral examples. Let's get straight into it.
All right, Section 1, Anatomy of a Virus. To really understand our microscopic adversary, we've got to look at its core building blocks, kind of building it out from the outside in.
Okay, so the outermost layer of many viruses is what we call the viral envelope. And you can really think of this as a biological invisibility cloak. It's actually a lipid membrane that the virus literally stole from a previous host cell, which lets it perfectly camouflage itself. Because it looks so much like our own cells, our immune system's security guards, well, they often just let it slip right by. This deceptive outer shield is really the virus's first line of defense, and unfortunately, the very first point of failure for our own immune response.
Now, just beneath that snaky envelope, we find the capsid. This is a tough, highly geometric protein shell, basically a microscopic suit of armor. It is meticulously designed to protect the virus's incredibly delicate core against some seriously hostile environments, whether it's battling acidic conditions, extreme temperatures, or just the harsh environment outside a human body. This geometric armor makes sure the payload actually survives long enough to reach its target. It's honestly a total masterpiece of evolutionary engineering. And inside that armor lies the absolute ultimate weapon, the genetic payload. We're talking RNA or DNA. This is the virus's raw blueprint. And what's truly wild here is that a virus doesn't actually have a life of its own, right? It can't generate energy or reproduce by itself. Instead, it just carries the exact malicious code it needs to hijack your cells and force them to do all the dirty work. This tiny, tiny strand of genetic material is literally the sole reason these microscopic entities can bring entire civilizations to a dead stop. Moving on to Section 2, Mechanisms of Infection. Now that we've sketched out the anatomy, let's look at the dynamic, step-by-step tactics these structures actually use to launch an attack. So, every viral invasion follows this incredibly ruthless three-step sequence. First up is cell entry. The virus physically breaches your cell wall, acting almost like a molecular lockpick, popping open a door it absolutely shouldn't have access to. Second is replication. Once it gets inside, it completely commandeers the host's own cellular machinery. It literally turns your cell into a relentless virus factory, just churning out copies of that malicious genetic material we just talked about. And finally, protein synthesis. The cell is forced to assemble a brand new army of pathogens until it eventually just bursts, releasing thousands of new viruses to go infect the rest of your body. Which brings us to Section 3, Metrics of Deadliness. We've mapped out the invasion, but how do we actually, objectively measure the true threat level of these microscopic machines? Well, epidemiologists evaluate a virus using three critical factors to calculate its destructive potential. First, there's the transmission rate. So exactly how fast and how easily does it spread from person to person? Second is the mortality rate, meaning what percentage of the people infected will unfortunately not survive. And third is mutation ability. Basically, how quickly can the virus genetically adapt to dodge our vaccines, our medical treatments, and our natural immune responses? And this leans to what is honestly a terrifying balancing act of biology. See, highly lethal viruses often burn out way too fast. If a pathogen has a massive mortality rate, it might actually kill its host before they ever get a chance to, say, walk onto an airplane and transmit it to 1,000 other people. But on the flip side, a highly transmissible virus might sweep through a massive population, but only cause really mild symptoms. The absolute most dangerous viruses in human history, they're the ones that find that perfect, devastating balance on this scale, spreading just enough before finally revealing their lethal nature. So, it begs the question, which pathogens have actually mastered this deadly architecture? Which viruses have successfully weaponized these microscopic traits to literally alter human history? And why should we care today?
Let's dive into Section 4, the deadliest viral examples. We're going to apply this whole framework we've mapped out to 5 of the most infamous real-world pathogens.
First up on the board, we have Ebola and Marburg. These are what we call filoviruses, named for their really distinct, almost thread-like shape. And they're absolutely infamous for causing severe hemorrhagic fevers. Their molecular structure is specifically engineered to attack the body's vascular system and immune cells at the exact same time. This triggers catastrophic internal bleeding, widespread inflammation, and just incredibly rapid systemic organ failure. It is a brutal, highly destructive microscopic interaction.
But what really makes these two stand out on our deadliness matrix is the sheer, terrifying weight of their mortality rates. Marburg, for example, has recorded absolutely staggering fatality rates of up to 88% in historical outbreaks. Now, they don't spread quite as easily as, say, a common airborne cold, but man, once they successfully breach a human cell, their lethality is practically unmatched in the natural world.
Next up, we have smallpox. Now, this features a highly complex DNA virus structure that basically achieved an epidemiologist's absolute worst-case scenario. It successfully weaponized both a high transmission rate and a high mortality rate. It was airborne, it was wildly contagious, and it ravaged global populations for literally centuries, fundamentally altering human demographics. It was so devastating, in fact, that humanity actually had to unite to fight back with an unprecedented global vaccine campaign. And because of that incredible effort, it remains the only human disease we have ever completely eradicated from the wild. Now, HIV operates completely differently. It's a retrovirus. So instead of just entering a cell and replicating normally, it uses this super insidious reverse engineering mechanism to permanently stitch its genetic payload directly into the host's own DNA. I mean, think of it like a malicious hacker completely rewriting the root code of your computer. And when you combine that with an off-the-charts mutation rate, it essentially just hides within the patient's own genome. This makes it incredibly difficult for the immune system to clear out, forcing us to develop highly specialized antiretroviral therapies just to keep the virus at bay. Finally, we look at COVID-19. This novel RNA virus utilized a very distinct spike protein, those microscopic molecular keys you often see illustrated, and they were specifically adapted for rapid cell entry right into the human respiratory tract. This specific architecture perfectly optimized the transmission rate factor. It spread incredibly efficiently, often completely asymptomatically, and it proved a really profound point. A virus absolutely doesn't need an 88% mortality rate to fundamentally shut down the modern globe. Its sheer transmissibility was its ultimate weapon. So, we've seen how envelopes, capsids, and genetic payloads can work in this devastating harmony to invade, replicate, and destroy. But here's the thing you have to remember. Nature is always experimenting. As these viruses continue to share traits and genetically adapt, we have to ask ourselves, what is the next evolutionary blueprint going to look like? And when the next molecular assassin inevitably arrives, will we actually be ready for it? Thanks so much for joining us on this explainer.